Welcome back if you were here yesterday. This is going to be, we're going to be talking about hypothesis testing. It's not an easy subject, but I'm going to basically focus on the fundamentals. I'm going to give you the foundation for you to understand hypothesis testing. So, sort of the outline is I'm going to talk to you about the fundamentals of hypothesis testing. This is going to be basically the biggest portion of the lecture, the fundamentals. And then I thought, I mean, there's so much to talk about hypothesis testing, so, you know, one has to pick what to talk about. I thought superiority versus non-inferiority versus equivalence. I'm going to talk a little bit about that, just to give you a sense. And multiple comparison is another one, which is a huge topic, but it's very, very important. I'm not going to go too much into the details, but at least make you aware about the issues. And then I'm going to give you a couple of slides just as a summary, bottom line summary of what, what was discussed throughout the lecture. So let's get started with the fundamentals of hypothesis testing. So I have a question. Now it's unfortunate because you have the answer. If you haven't looked the answer, don't look. If you have looked at the answer, don't answer. Without blank, there is no need for statistics. Without blank. So what I usually get, I mean, this is a little bit difficult to have too much interaction with all the videos and the cameras and but what I usually get is without data, and that certainly is a good answer. But the key thing is that without variability, there is no need for statistics. So you're going to say, what? What is he talking about? So let me show you. It's an illustration. So you do an experiment, you have a clinical trial, and the average of the old treatment is on the left, the average of the new treatment is on the right. High numbers are good, so high is good. So, and I tell you, here's the average of the old treatment and the average of the new treatment. The question is the new treatment better than the old treatment? Yes or no? What do you think? Well, I mean, certainly the average of the new treatment is higher, but you need to know a little bit more about the spread of the data. Without the spread of the data, you don't have a good sense. Let me illustrate. If I told you the data looks like this, then you say, oh yeah, well then definitely the new treatment is better than the old treatment. Because now you have a sense of the variability. And all the white points are to the right of the green points. So we're comparing the variability within a treatment, within each treatment, compared to the overall variability. That's what basically we're doing in the back of our minds. So here you would say, yes, the new treatment is better than the old treatment. Now, if I spread things around, I would say, yeah, still, the new treatment is better than the old treatment. Now, what if I do some more? So now you say, well, I don't know. I see some white dots to the left, and I see some green dots to the right. So it becomes a little bit, you have to think about it, and you ever wonder why we call it analysis of variance when we are really interested in comparing means? Why do we call it analysis of variance? Well, because we're analyzing variance in order to do inference on the means. But we're analyzing variability. And that's what this is about. I mean, you look at this and you say, well, how does the variability within a treatment compares to the overall variability of the whole data? By the way, the dots don't really average to the average. I just put them like that. So if you want to calculate, that's probably not going to produce the average that you see. So it's just to illustrate, make the point. All right. So statistical inference, what do we mean by statistical inference? We draw a sample from the population of interest. It's a procedure in order to draw conclusions. So we draw a sample from the population of interest. We analyze the data from the sample, and we make conclusion about the population based on results from the sample. That is statistical inference. Okay, let's go a little bit deeper. So what is the typical setting of statistical inference? Statistical inference and hypothesis testing, they're kind of the same thing. Okay, so what is the setting? Well, I'm going to show you the setting for non-Bayesian. And I'll talk more about it. But for now, just keep in mind, this is non-Bayesian setting. There is a null hypothesis we call H0. We label it as H0. We call it the null hypothesis. And we're basically saying the experimental is equal to the control, or the difference between experimental and control is 0. So experimental may be a new medication, control may be placebo, and we're saying that the new medication and the placebo have equal effectiveness. They're equally effective. Or the difference in effectiveness of the two is 0. That's called the null hypothesis. I mean, this is a typical setting. Not every single hypothesis testing looks like this, but that's sort of a typical one. And then we have an alternative hypothesis. We label it H1 or HA, H0, H1, or sometimes A for alternative. And we're basically saying the experimental is different from the control, or the difference between experimental and control is different from 0. So again, the new medication is really different from the placebo, hopefully more efficacious, but that's basically the alternative hypothesis. So what's the question is? The question is there enough evidence to reject H0, the hypothesis of no difference? So we would like to reject H0. This is what you set the null hypothesis with the expectation and the hope that you're going to reject it in favor of the alternative hypothesis. Okay? Are you with me? Please nod your heads. Okay, thank you. Okay, so this is you may have seen it. It's a very typical table that is shown in all the intro textbooks to hypothesis testing, but it's a very important table. Very important table. So here you're doing the experiment and you're going to make a conclusion based on the sample. Okay. And you're going to do two possible conclusions. You're going to say, I reject H0. H0 is the hypothesis of no difference. So I reject H0. I have evidence of a difference. Or I'm going to say, I cannot reject H0. I have no evidence of a difference. Okay? H0 is the hypothesis of no difference. Okay? So that's what the conclusion you're going to make based on your analysis, based on the sample. But then there is the true unknown state. Nobody knows it. We don't know what it is. But there is such a thing as the truth. Okay? And the truth could be either there is no difference, so H0 is true, the null hypothesis is true, there is no difference, or there is a difference, or H0 is false. Okay, so these are the two possibilities for the truth. We don't know the truth, but these are the two possibilities. There's the conclusion from our study, and there is the truth. So now let's see what are the possibilities, what could happen. There are 4 things that could happen. We could either make the correct conclusion, because we are rejecting H0. We say there is an evidence of a difference, and in fact, there is a difference. So we reject H0 when H0 is actually false, so we're making a correct conclusion. and we call it true positive. Why true positive? It's positive, there is a difference, and it's the truth. It's actually, there is a difference. So we call it true positive. Okay? Or we can make another way of making a correct conclusion is we fail to reject H0. We say there is no evidence of a difference, when in fact there is no difference. So we fail to reject H0 when actually H0 is true. So we're making a correct conclusion. We call it a true negative. Because it's negative, no difference, and it's true. Okay? So far, so good? Okay. Now, that's when we get into the statistics. We say if we reject H0 when actually there is no difference, we reject H0 when H0 is true, we claim a difference when there is no difference, we're making a mistake. We're making an error. We call it the type 1 error. It's a false positive. We're saying it's positive, but it's not correct. It's a false positive. It's a type 1 error. And then there is the type 2 error, what we call a type 2 error. So we fail to reject H0. We say there's no evidence of a difference, when in fact there is a difference. That's a mistake. That's an incorrect conclusion. It's a type 2 error. We call it the false negative, because we say it's negative when in fact it's not correct. Okay? So that's the sort of the setting where, what are the possible correct conclusions and the errors. And the two errors are different. So we call it the type 1 error and a type 2 error. All right? Okay. So now I'm going to get a little bit into stat language with alpha. I try to avoid Greek letters, but alpha is so commonly used, I can't avoid that one. So alpha is the probability of making a type 1 error. That's called alpha. It's the probability of making a type 1 error. It's the chance of actually rejecting H0 when it is true. It's the chance of claiming a difference when in fact there is no difference. And beta is the probability of making a type 2 error. It's the chance of failing to reject H0 when H0 is false. It's a type 2 error. And beta is the probability of making that error. Okay? All right. So I'm going to get a little bit deeper. So the alpha, like we said, is the probability of making a type 1 error. Sort of a non-technical definition for superiority trials, and we'll talk about this. It's the chance of concluding that the experimental treatment is more effective when in fact it is not. We're rejecting H0 when H0 is true. And that's the technical definition. It's the probability of rejecting H0, claiming there is a difference, when in fact there is no difference. And I just want to talk to you a little bit about the different perspective, like a regulatory agency versus a pharmaceutical company. So a regulatory agency, FDA, for example, find this as a very important parameter to control. The FDA does not want to approve a medication that's not efficacious or effective. So it wants to make sure that this kind of error is not made. Of course, it can't be 100% sure, but at least it wants to be able to say, if you, pharmaceutical company, you're going to make a clinical trial, I want this alpha to be this much. Because it's very important to the FDA not to approve a medication that's not efficacious. That's the FDA perspective. But the pharmaceutical company, not that they don't care, but, they're not going to really be devastated if their medication ends up in the market and in actuality it's not efficacious. So type 1 error, alpha, probability of making type 1 error, is very important to the FDA. To the pharmaceutical company, less so. And the most commonly used value of alpha is 0.05 for two-sided. It's the most commonly used. So beta is the chance, we said, of claiming no difference when a different exists. It's a mistake. It's an error. It's claiming no difference when a different exists. It's the probability of not rejecting H0 when H0 is false. H0 is the hypothesis of no difference. We're not rejecting it when, in fact, there is a difference. So since beta is the probability of making an error, a low beta is a good thing. Power is 1 minus beta. It's the probability of rejecting H0 when H0 is false. It's claiming there is a difference when there is a difference. We say also it's the probability of detecting an effect when it exists. So it's the right thing, it's a good thing, and so high power is good. But it's 1 minus beta. And just like I did with alpha, the non-technical definition of power is the chance of concluding that the experimental treatment is more effective when in fact it is. That's the power. The technical definition is the probability of rejecting H0 when H0 is false. So we're making the right conclusion. And just like with alpha, there are different perspectives. The FDA, now we're talking about putting in the market, not putting in the market a medication that is efficacious. So the FDA is not so much concerned about that. Doesn't get to the, I mean, of course they care about public health and they want to, approve medications that are going to cure diseases. But the pharmaceutical company, I mean, think about what we're talking about here, is imagine a pharmaceutical company who has in its hands and figured out an efficacious, an actually, truly efficacious medication, but then finds out through a clinical trial that, it doesn't show, it doesn't reject H0, doesn't claim a difference. It wouldn't like that. It has an efficacious medication in its hands and can't show that it is efficacious. That, we're talking about years and years of research, billions of dollars spent, and then it doesn't make it. When in fact, it is more efficacious than placebo, for example. So the point is, the power is very important to the pharmaceutical company. And alpha is very important to FDA. So it's nice because then both get controlled. So the most commonly used value for power is for early phase trials is somewhere between 0.6 and 0.8, 60% or 80%. And for late phase trials, meaning like further down in the research stream, it's between 0.8 and 0.95. Okay? So we've done a lot of technical stuff. If you don't change the sample size, if you want to keep the sample size the same, if alpha goes down, beta is going to go up. Well, let me first talk about these two. Alpha is the probability of rejecting H0 when H0 is true. So it is, basically it's an error. You want to make it harder to find a difference, to claim a difference. If you bring alpha down, you're making it harder to claim a difference. Well, if you're making it harder to claim a difference, you're making it harder to claim a difference when there is a difference. So that's why the beta, that error, goes up. So they're going in opposite direction. If you're going to make it easier to claim a difference, then you're going to make an error of claiming a difference when there is no difference. So alpha goes up. But then your beta goes down, because you're going to make it easier to find a difference when there is actually a difference. So they're going in opposite direction, and because power is 1 minus beta, whatever happens to beta, the opposite happens to power. So if alpha goes down, beta goes up, power goes down, without changing sample size. If you change sample size, that's a different story. Okay, so let's do something a little bit more fun here. And we're going to do an experiment. And I'm going to talk about this experiment through several slides, so stay with me. We're going to do an experiment. I have a magician's kit. Okay, I have a magician's kit. In that kit, there is a coin that it's a regular coin, has a head on one side and a tail on the other side. But I also have a coin that has 2 heads, a head on each side. Okay? So I'm going to take these 2 coins, put them in my hand, and I'm going to show 2 hands, both hands to you, and you're going to pick a hand. Okay? So you're picking one of the 2 coins. We don't know which one you pick. I don't know which one you pick. You don't know which one you pick. But I'm going to toss it, even without looking, and I'm going to look at the results. So we have 2 coins. One has tail and head, and one has 2 heads. And we're going to toss it. Okay, and I'm going to give you the data. And I'm going to ask you to decide which coin am I tossing. Okay? So the null hypothesis is that it's the regular coin with head on one side and tail on the other side. That's the null hypothesis. It's the regular coin. And the alternative hypothesis is that it's the other coin. It's the one that has head on both sides. And we have equipoise. I talked about it yesterday, about equipoise is, it could be one or the other. We don't know. It's equally likely that we have the regular coin as it is to have the 2-headed coin. Okay? So I'm going to put this sort of in the corner. There's the experiment. The hypothesis is the null hypothesis. It's a regular coin. The alternative hypothesis is the coin that has 2 heads. So I'm going to toss the coin. I'm going to give you the data. Now, what I didn't say is that you're going to decide which coin am I tossing. But this is a serious decision. It has major public health implications. Okay, so your decision is very important. You're going to have to decide which coin it is. Okay, let's make it even more concrete. You're going to pay me $100 for each toss, and then you're going to stop because it costs you to toss each time. It's like it costs to do clinical trials. We could do 1,000 clinical trials, but we can't afford it. So it's going to cost you for me to toss a coin every time. It costs you $100. But then you're going to tell me, stop, that's enough. I'm ready to make a conclusion. And you say, okay, this is the coin you have. And if you're right, I'll give you $1,000. And if you're wrong, you give me $1,000. Okay? So basically I'm saying your decision is very important, and you can't say, oh, let's continue to do it, a zillion times, and then I'll tell you. That's basically the setting I'm trying to put. Okay. So I toss it once, and I get a head. So my question is, are you ready with this amount of data to reject H0? In other words, to say, oh yeah, that's the 2-headed coin. You got the head, it must be the 2-headed coin. Are you ready? Would you say at this point you're ready to make a decision? No. I would think most of you would say, or all of you would say, no, not yet. Okay. So now I'm tossing it twice, and we get 2 heads. 2 heads. Are you ready now to say, I am ready to conclude that we have the two-headed coin. You, Paul, are tossing the two-headed coin. No? No, I see a lot of heads says no. Okay. So you're not ready to reject H0. What about 3? 3 tosses, 3 heads. No? Say that again? Is it an early or late stage trial? Good question. It's a tossing of a coin. But it has major public health implications. Your decision is very important. You have to decide which coin are we tossing. Are you ready to say, okay, well, 3 heads out of 3? I would say, yeah, you must have the two-headed coin. Would you be ready to say that? Well, keep it. You tell me. You decide in your head, and I'm going to show you. What about 4 heads, 4 tosses, 4 heads? Think about it. Are you ready to say, yeah, at this point, the lady over there says she's ready. She would be ready to say, Paul, you're tossing the two-headed coin. Okay. 5 tosses, 5 heads? Yeah, I see some heads would say yes. 6 tosses, 6 heads? Definitely. 7 tosses, 7 heads. Is there anybody who would still not conclude, who still would want maybe one more toss? Yeah, okay. One person would want to see one more toss. Okay. Okay, another one. So keep in mind, just think about where, this is kind of interesting, where did you say, or where would you say you would stop and ready to make a conclusion? Just take 5 seconds in your head, think about where, you're not going to tell me, just keep it in your head, because I'm going to show you something. Where would you stop and say, okay, I'm ready to conclude that I have a two-headed coin? Okay? All right. So, here are the p-values. So what are these p-values? We haven't talked about p-values. Well, the p-value is the probability of getting, let's talk about the very first one. The probability of getting ahead when I'm actually have the regular coin. It's the probability of getting the data I got, which is ahead, when H0 is true, when I am actually tossing a regular coin. What is the chance of getting ahead when I'm tossing the regular coin? It's a half, right? It's 1/2. There is a 1/2 chance. If I'm tossing the regular coin, there's 1/2 chance of getting head. Okay, what is the chance of getting 2 heads in a row if I were tossing the regular coin? It's 0.25. It's half times half. 3 in a row, 0.125. 4 in a row, 0.063. That's the p-value. That's the p-value. It's the chance of getting the data you got under the null hypothesis if you were actually tossing the regular coin. Yes? We're tossing one coin, either this one or that one. We don't know which one. But the p-value, the definition of the p-value, it says if you were under the null hypothesis. So in this case, if you were tossing the regular coin, that's the definition of the p-value. If you were tossing the regular coin, what is the chance of getting 2 heads? What is the chance of getting 3 heads? And if you go to 7, what is the chance of getting 7 heads if you are actually tossing a regular coin? That's the definition of a p-value. And it's 0.008. So it is very, very low, right? So think with me. I mean, so I did the data. I tossed the coin 7 times, and I got 7 heads. That's the data. I can't say, oh, no, you didn't. That's facts. But it has a very low chance of happening if H0 were true. But I can't change the facts. The fact is a fact. The data are the data. So what am I going to be suspicious of? What am I going to question? What else is there to question? The hypothesis, right? Because it says, if the null hypothesis were true, the chance of getting what I got is very, very low. Well, what I got is what I got. So what else could be wrong is the hypothesis. So therefore, I'm going to reject H0. Because the chance of getting something, what I got, under the null hypothesis is so low that it couldn't be what's going on. And that's the p-value. So, how many consecutive, you can answer to yourself, how many consecutive, where did you decide to reject H0? And how does this compare to the p-value to the alpha of 0.05? So you decided to stop at 4. Well, you decided basically with a p-value of 0.063. And it's a tiny bit higher than the regular commonly used 0.05. So 0.05 is used as a cutoff to say, okay, if the probability of getting what we got under the null hypothesis is lower than 0.05, we reject the null hypothesis. So if you understand what I just said, you're basically understanding the foundation of hypothesis testing, the non-Bayesian. Okay? So I'm kind of summarizing at the top of this slide. We have the 2 hypotheses. Null hypothesis is a regular coin. Alternative is the 2-headed coin. We did an experiment. We have 7 tosses. The data, we got 7 heads. P-value is 0.008, right? It's from right here, 0.008. And the conclusion is because it's below 0.05, we say we reject H0. Okay? So, now, have we proved the alternative hypothesis? In other words, in English, have we shown for sure that we must be tossing the 2-headed coin? Have we proved that? No, we haven't proved it. We haven't shown that it is the 2-headed coin for sure. We are pretty confident that it is the 2-headed coin, but we haven't shown it. We haven't proved it. So the answer is no. Is 0.008 the likelihood that the results are due to chance? And the answer is no. It is the chance of getting what you got. Is 0.008 the probability that H0 is true? No. It's not the probability that H0 is true. Is 1 minus 0.008, 0.992, the probability that the alternative is true? No. It's just simply 0.08 is the probability of getting 7 heads if it were a regular coin. Not more, not less. What I've shown you on the slide is the commonly misinterpreted p-value, the way p-value is misinterpreted. You're not proving anything. You're not saying this is the chance that it is true or the chance that it is false. You're just saying this is the chance of getting what I got if the null hypothesis were true. So now we can really just repeat what I just said, which is the definition of a p-value. The p-value is the probability of obtaining a result as extreme or more extreme than the one obtained if H0 were actually true. And if the p-value is less than or equal to alpha, usually 0.05, we reject H0. We say, you know, the chance of getting what we got under the null hypothesis is low enough that I'm confident that H0 cannot be true, and therefore I'm going to reject H0. I'm going to reject the hypothesis of no difference. And if p-value is greater than alpha, I say, well, it could happen. H0 could be true. Because I got data, and under H0, it is possible to happen. So I'm not rejecting H0. And like, you know, the commonly used alpha levels is 0.05 or sometimes 0.01. Okay. Now let's talk about Bayesian. Now, if I was giving this lecture, I don't know, maybe 5 years ago, I wouldn't talk about Bayesian statistics because I would be concerned that it's going to confuse you more than it's going to help. But today, I feel like, you know, it's like burying our heads in the sand if we don't talk about it. Because it's gaining a popularity, it's gaining use, a lot more people using it. The FDA has embraced it. So just to not talk about it, it's like, you know, ignoring something that is going on. So even though I'm still concerned that I'm going to confuse you more than help you, I feel like, you know, this is a topic I really need to talk to you about. Bayesian stat is not new. It's been around for a long, long time. But because of the power of computer, it's been easier and easier to actually program and get results. And so that's how it gained a lot of popularity. And more people are using it. So I have to tell you about it. And I'm going to tell you about it. It's very intuitive. Actually, I think, personally, and I know statisticians get very excited about Bayesian versus non-Bayesian. So when you get a statistician who's a Bayesian with we call it a frequentist or a classical statistic, they really get it, enthusiastic. So I personally find it as very intuitive. And here's the thinking, here's the reasoning behind the Bayesian approach. It says basically you start, before you do anything, you must have some prior belief. You must know something, okay, about whatever you're measuring. Okay, so you have some idea. And then you do the experiment and you get data. And basically what you're doing is you have posterior belief. You're updating your belief based on the data. So you have a prior belief, you have the data, and you put the two together, and now you update your prior belief, and we call it posterior belief. Posterior is after the experiment, and prior is before the experiment. Okay? So you have some idea, then you do the experiment, have the data, and now you have the posterior belief. But then the posterior belief becomes the prior belief for the next experiment. So the posterior now becomes the prior for the next experiment. You get some data, and you update the information. And that's how we actually live, right? I mean, you live, you're born, and you slowly learn, and, you know, you think something, and then something happens, and you adjust, you update, and then you keep updating as you live. And that's basically what this is doing in a much more formal way, the reason it's called Bayesian is because it's using Bayes' rule. Bayes was I don't know, not a minister, I forgot, but basically he came up with the rule. It's about conditional probabilities. So there is a Bayes rule, and the whole Bayesian approach is based on that rule. So So now the posterior belief after 2 experiments becomes the prior belief. And you get data, and you update that information. And then you keep on doing it until it's pretty clear, whether the medication is efficacious or not. So there is enough posterior belief to conclude efficacy or futility. Or even you can say, I have enough evidence now that there can't be any difference between the medication and placebo. But it's done sequentially. So that's kind of just the thinking behind it. The math and the stat gets very complicated and very theoretical, but just the thinking. So now let's go back to that tossing of coin and the whole experiment we did. The Bayesian don't think about p-values and alphas and all that. They say, well, we're starting with 2 possibilities. It's a regular coin or it's the coin has 2 heads. And up front, I have, because of equipoise, there is half a chance that it's the regular coin and half a chance, 0.5 chance, that it is the 2-headed coin. Right? At the beginning, I haven't seen anything. I haven't seen any data. That's what I think. It's 1/2 chance it's a regular coin and 1/2 chance it's the 2-headed coin. Or you can say that the probability of a head being 1/2 has itself 1/2 chance, and the probability of the head being 1 equal to 1 has itself 1/2 chance. So calling it regular coin is the same as saying the probability of a head is 1/2. Calling it a 2-headed coin is the same as saying that the probability of head is 1. So if we start with the prior belief that H0 and HA, the alternative, are equally likely, which because of, we said, it was a same equal chance. So this is, I hope you can read it, because I'm going to put lots of these little squares on the slide, so I had to make it small. But basically it says, think about, if this is confusing you, replace the p equal to 1/2 to regular coin. Put the label in your mind, regular coin. Actually, I was thinking of doing this, changing it at the last minute. But put here regular coin, 2-headed coin. Okay? So when we start, the prior is, it's equally likely to be the regular coin as to be the 2-headed coin. And then we do the toss. So we're going to update this distribution. We do a toss, and we find head. Now I'm going to update this information. And now, because I got a head, the probability that it is the 2-headed coin has gone up, and the probability that it is the regular coin has gone down. And then I do another toss, and it's a head. So I'm updating the chance that it is the regular coin, and I'm updating the chance that it is the 2-headed coin. And you can see it goes up for the 2-headed, it goes down for the regular coin. And you keep on doing this, and you keep updating. Again, prior plus data becomes posterior. So this one was prior, and this one was posterior for this experiment. But for this experiment, the posterior became prior. Then you do the experiment, then you update, and it becomes posterior. But this posterior is the prior for this experiment, and so on. So you keep on doing it, and after 7 tosses, the chance that it is the 2-headed coin is really high, and the chance that it is the regular coin is really low. But note, and that's where, the confusion. When we were talking about the classical stat, I said it's not the probability that H0 is true. It's not the probability, when we're talking about p-value, it's not the probability that the alternative is true. That's when I was talking about classical. When I'm talking about Bayesian, I'm actually saying this is the probability it's the 2-headed coin, and this is the probability that it is the regular coin. So you're saying, oh, my guys, decide which one is it? Well, it depends on which approach you're using. So after 7 tosses and 7 heads, our updated belief after 7, so we're talking about the very last graph, is that we are 0.8% sure that we're talking about we have the regular coin, and we are 99.2% sure that it is the two-headed coin. Now, I just want to make sure, well, I'll show you in the next slide, but I will say it right now. These numbers are not exactly the same as, they're not the p-values, okay? They were calculated differently, okay? This is very important. I mean, yes, we're saying, you know, we're stating conclusions in a very different way, the classical and the Bayesian, but the numbers are not the exact same way. And I'm going to show you the slide where the two are compared. So here's the Bayesian posterior probability after each toss. So what I'm showing you here is the Bayesian, the probability of a regular coin, of tossing a regular coin after each toss. And you can see that it's going down after each toss because we keep on getting heads. And this is where it compares to the p-values, the frequentist p-values. They're not equal, but very interestingly, very, very close. The one that is the farthest is after the one toss. After one toss, the Bayesians say, oh, you know, there is a 1/3 chance that you are tossing the two-headed, the regular coin. But the p-value goes down to 0.5. But after that, they get pretty close. So again, these numbers and these numbers are calculated completely differently. They are also interpreted completely different. We said these p-values are not the probability that it is the regular coin, are not the probability that it is the two-headed coin. And here we are saying 0.08 is the probability that it is the regular coin. So again, if I've confused you, I had to talk to you about Bayesian stat. So, did somebody have a question? I don't know if okay, no, I guess not. Okay. So what is the connection between alpha and p-value? If the p-value is less than alpha, typically 0.05, the null hypothesis of no difference is rejected, and the result is declared statistically significant at the 5% alpha level. So you could compare p-value to the alpha level that was set up front. And if it is less, then you call it significant. If it is more, you call it not statistically significant. What is the connection between p-value and sample size? Okay, so let's talk a little bit about that. The connection between p-value and sample size. So we have a randomized control trial, and we compare two treatments. And we collect the data, we do the experiment, collect the data, analyze the data. The analysis is done, and we get a p-value of 0-something. Okay? And we're going to talk about this. So this is the distribution of the difference between the two treatment groups, if in fact there is no difference. So you're saying, well, wait a second, I'm confused here. If there is no difference, why isn't it just 0? Why do we have a distribution of the difference? Because it's basically saying if you were to run the trial 100 times, the difference under the hypothesis of no difference between the two treatments, there's still going to be some differences. You're going to get different differences depending on the trials. So each trial, you're going to get one number. You're not going to get exactly 0. So this is the distribution of the difference. But here's the point. The point is you observe a treatment effect of 3. So the difference between treatment and placebo is 3. And you want to calculate the p-value. So the p-value is the probability of getting a difference as extreme or more extreme than the one you got. Under this distribution, the one-sided p-value is 0.16. One-sided because you only look at, you only look at whether it is greater, and that's it. And the two-sided is you look at both sides. It could be more extreme this way, or it could have been more extreme the other way. So the two-sided p-value is 0.32. And therefore, or the confidence interval is shown here. It's the treatment effect is 3, but the confidence interval covers 0, and basically the p-value is greater than 0.05. Bottom line, you don't reject H0. You claim no difference between the two treatments. Okay? I can't hear you. It's coming from this distribution, which you can calculate. So it's the distribution of the difference under the null. Once you get this distribution, and you look at the observed treatment effect, which is 3, and then you look at the normal table with this, the mean 0 and the standard deviation or the standard error of the difference, whatever you got. And so you have this distribution, and you look it up in a normal table to get the area under the curve away from 3. And nowadays we don't go to books and look in a table. We let the computer tell us. I mean, Excel does these things, and that's where the 0.16. It's this area is 0.16. And when you look at both sides, it's the double. And that's the definition of a p-value. Okay, so now what we do is I'm going to keep everything the same. We're going to look at this. We get the same treatment effect, same everything, just the sample size is increased. So same variability of the data and same treatment effect, 3. But because the sample size has increased, the standard error of the difference, the variability, you notice the curve has gotten thinner and higher. And that's because the variance of this curve is the standard error of the difference. And the standard error of the difference has a square root of n in the denominator. So as the sample size goes up, the standard error goes down and the curve gets tighter. I'll tell you what the point of all this is at the end, but just stay with me on this one. So the treatment effect is still 3, and I look at the p-value. Now the p-value, same idea, is 0.067. 0.067, again, is this area under the curve, away from 3, the treatment effect that we found from our experiment. And the two-sided p-value is 0.134, which is double the one-sided. So I haven't done anything different. The only thing different between the two is that I've increased the sample size. It's the same treatment effect. It's the same variability of the data. But the standard error of the difference, because it has square root of n in the denominator, has gone down, and therefore the distribution has gotten tighter. So still it's above 0.05, and I claim that I do not reject H0. And there's the confidence interval for the difference. And again, here I'm going to do the same thing. I'm going to increase n some more. And with exactly the same treatment effect of 3. Because I've increased the sample size, standard error of the difference goes down, and the curve gets tighter. Now when I get p-value, I get a tiny, tiny area, which is 0.0013. And if I look at the two-sided, I get 0.0026. So now I have a statistically significant result. So you're going to say, but wait a second. I mean, the treatment effect is the same. It wasn't statistically significant the first slide, and now it is highly statistically significant. Well, yes, that's because of the sample size. So the point is, the sample size can make a huge difference in terms of finding something statistically significant. The key question is, even if you find something that is statistically significant, is it clinically meaningful? You have to ask yourself, is 3 a clinically meaningful difference? So we always say statistical significance does not mean clinical significance. Okay? So why am I showing you all this? Is because this is where power analysis comes in. You ask, the statistician will ask the clinician or the researcher or the investigator is going to say, if we were to find a difference of 3, would you call this 3 a clinically meaningful difference? And if the investigator says yes, then let's make sure that if the difference of 3 is what we get, that we will be claiming it to be statistically significant. How do we ensure this? With the sample size. Of course, if we make the sample size 1 million, everything is statistically significant, but we can't afford that. So we want the smallest sample size that will find a statistically significant result if the difference was actually 3. We would be claiming it as statistically significant. And I just told you in a couple of sentences the whole idea behind power analysis. So, what's the point? The point is there are 2 ways to get statistically significant results guaranteed. You know, it sounds like a commercial. Guaranteed. If you analyze a very large sample, you are guaranteed to find a statistically significant. If you make it large enough, it's going to be statistically significant, regardless of the results. Well, unless it's going the opposite way, unless placebo is better than the medication. Okay? But the tiniest difference, you can make it statistically significant if you increase. But you can't afford. So that's where we do power analysis. What is the minimum sample size that will make 3 look statistically significant? I'll tell you the second way later. So, what is the connection between confidence intervals and hypothesis testing? I haven't talked about confidence. I have sort of mentioned confidence intervals, but I really haven't talked about it. So a 95% confidence interval looks like this. It has the point estimate. We say the point estimate or the estimated mean or the average. We call it the point estimate. So that's the number we got from the data. But then we calculate a lower limit and an upper limit. Okay? And here's the interpretation. The natural perspective says that there's a 95% chance that the true unknown value is inside the confidence interval. So in other words, we are 95% confident that the true unknown value is somewhere within the confidence interval. Different way of saying the same thing. So if the confidence interval is this thing right there, and the true unknown value, we just don't know where it is, we think it has a 95% chance to be inside the confidence interval. Okay, were you impressed with the animation? Okay. That's the natural perspective. But the frequentist, sort of, if you want to be pure about it, they would say there is a 95% chance that the confidence interval covers the true unknown value. So basically the frequentists say, well, the true unknown value doesn't move. The true unknown value is fixed. We just don't know it, but it's fixed. And it's the confidence interval that moves, because if you do different clinical trials, you're going to get different confidence intervals. So they say, here's the true unknown value, and it is the confidence interval that either covers the true unknown value or does not. So, I find that it's, splitting hair. So just the idea is that you're 95% confident that, the true is inside this confidence interval. You may have seen this. If you were to run the clinical trial 100 times and you calculate each time 95% confidence intervals, so you have 100 95% confidence intervals, 95 of them are going to cover the true value and 5 are not, overall, roughly, not exactly, roughly. That's basically another way of looking at it. If you were to do it 100 times, 95% of the time, you would have included the true value. And by the way, don't count the lines because they don't add up to 100. But I couldn't put them all on this graph. But the 5 green ones, yeah, are definitely clear. So 5 are out, do not cover, and 95 do cover. What is the connection between confidence interval and hypothesis testing? If the 95% confidence interval does not include the value of the null hypothesis of 0 difference, the result is statistically significant at the 5% level. So if you have a confidence interval and your null hypothesis is that the difference is 0 and the confidence interval for the difference doesn't cover 0, you have a statistically significant result. And if it does cover 0, you don't have a statistically significant result. That's the connection between the two. So if it does, the result is not, if it does cover the 0 difference, it is not statistically significant at the 5% level. So they're very, very closely related, confidence interval and hypothesis testing. So that's the fundamentals. 1 hour, okay? It's not easy. You have to get used to it, and but that's the fundamentals. So let's talk about superiority, non-inferiority, and equivalence. So let's start with superiority. So the clinical hypothesis is that the experimental treatment is more effective than the control treatment. So you want to you have a medication and you want to show that it's better than placebo. You do a superiority trial. Or you want to show it's better than treatment as usual. Or you want to show that a medication is more efficacious or effective than psychosocial therapy. You do a superiority trial. So technically speaking, the null hypothesis is that the experimental is the same as the control, is equal, the effectiveness are the same, and the alternative hypothesis is that they are different. You're going to say, but wait a second, I thought we were talking about superiority, and here we say it's different, so it could go either way. Well, you're hoping that you're going to show that the medication is better than placebo, but because you're not sure and you don't know and you have equipoise, you allow for going either way. And that's why the hypotheses are set as equal or not equal. And as always, we set the null hypothesis with the hope, with the expectation of rejecting it in favor of the alternative. You set the null hypothesis with the expectation or the hope to reject it. You want to reject the null hypothesis. You want a p-value that's less than 0.05, so that you say, I reject the null hypothesis. So, in a graphical way, so let's say the difference is 0. This is the vertical line. And so I'm going to show you 95% confidence interval around the difference from the data, from the experiment. So I'm showing you experimental minus control. And high numbers on the right, represent good outcome. So if the difference is on this side, it means the experimental is better than the control. Okay? So you find the confidence interval from your data, and this is the difference with the lower limit and upper limit. And the whole confidence interval does not cover the difference 0, so you claim superiority. The difference is statistically greater than 0. So you have a statistically significant result. If it's on the other side, you actually have inferiority. You have, because the difference is on the left side of 0 completely. The whole interval is on the left side. So you know that the control is better than the experimental. The placebo is better than the medication. I've never seen a result like that, but theoretically. And if it covers 0, then you do not reject H0. So here are if you're on this side, you're going to reject H0. If you're on this side, you're going to reject H0. But if it covers 0, you do not reject H0. That's the conclusion. So what about non-inferiority? So in the clinical hypothesis that the experimental treatment is not less effective than the control treatment. It's not less effective. Okay? It's not worse. It's about as good as. In technical terms, the statistical hypotheses are that the experimental is less than the control minus a delta. In other words, you say, I can go lower than the control, but up to a delta. If I still stay within this difference delta, which is called the non-inferiority, the margin of non-inferiority, if I'm still within the non-inferiority margin, I'm okay. It's not worse. But if I exceed that, then I say it's worse. And I'll show you. And again, as always, we hope and expect to reject H0 in favor of the alternative. So now I've added a new thing. Not only I have 0, but I have the non-inferiority margin, which is minus delta. Same thing. High numbers on the right are good. So if my confidence interval for the difference is on this side of 0, I basically have a superior. The experimental is superior to control. And if it's on this side of minus delta, it's inferior. And here's the key thing. If the confidence interval is to the right of this minus delta, that's when I claim non-inferiority. Because I'm saying, yeah, it may be less than 0, but it is within the margin. And I'm completely within the margin. So that's non-inferiority. If it covers either one, it's inconclusive. If it covers the minus delta, inconclusive. If it is here, because it's completely to the right of minus delta, it is non-inferior. That's the conclusion. The technical conclusion is non-inferior. The reason I put a question mark here is because, well, it's non-inferior, but it is completely to the left of 0. But technically speaking, you have shown that it is not inferior because it is completely to the right of the minus delta. If it covers the minus delta, then you can't say much. And here's basically, if you notice here, if the lower limit of the interval is to the right of minus delta, as long as you're completely on this side, you reject H0. You say it's non-inferior. H0 says that it is inferior, and so you reject it. And if you are the other three possibilities, you do not reject H0. And equivalence basically says if you want to compare two medications that are out there, maybe one is expensive, one is cheap, maybe you have a generic, and you just want to show that they are just about the same. So you talk about equivalence. So here's a fancy way of saying the null hypothesis is one is better than the other, and the alternative hypothesis says is they're about the same. They are within this margin of delta. And as usual, you hope and expect to reject H0, the null hypothesis, in favor of the alternative. So now you have both the minus delta and the plus delta. And as long as you are within these limits, you're going to claim equivalence. If you start getting out of these limits, you cannot conclude, you do not reject H0. So here it's equivalent. Here, I'm sorry? Very good question. The question is, how do you come up with the delta? It's very hard. It's not a statistician who's going to come up with this delta. It's the clinician, the subject matter experts. And that's why some people don't like equivalence and non-inferiority trial, because they say, well, I don't know, I have to pick a delta. I don't know what the delta is. And you can see that the selection of the delta is key. So it's not trivial, but if you want to do non-inferiority, you have to decide. You have the subject matter expert has to say, ask themselves and say, if the difference can go all the way to 3, and I still think they're about the same, but more than 3, boy, it's this. Well, I was going to say it's the same when you do power analysis. You have to say, what is the difference that you would claim as clinically meaningful? Well, again, it's not easy, but it's a question you have to ask yourself. So, yeah, thanks for the question. So again, it's not a statistician who's going to come up with a delta. I mean, a statistician doesn't know the subject matter as well as the subject matter expert. So the clinician. So if you are within these two limits, you're going to call it equivalent. But as long as you get out of them, then you do not reject H0. So again, as long as you are within, you reject H0, meaning you claim equivalence. And if you're outside, or if you cover any one of them, then you cannot reject H0. Okay? I'm going through these things. So, every slide I show you has, I don't know, dozens of books on the topic and thousands of papers on the topic. And so we're just, going over, the very tip of the topic. Okay, so multiple comparison. As I was saying, I'm not going to go in details. This is another one that is, very, very complex. There are experts just in this, in multiple comparison. There are statisticians that's all they do is adjusting for multiple comparison. I'll explain what this is all about. But it's very important, and it's very easy to ignore it. So I'm basically saying, look, there is this thing. So when you go back to your work, maybe you won't remember the details, but you'll remember, oh, wait a second, aren't we supposed to do multiple comparison adjustment? So what it is. And the first analogy, I heard it from Michael Proshan. He's a biostatistician at the NIAID. And that's what I heard it from the first time. It's not exactly the way he says it, but I kind of made a variation of it. So you have a dartboard, and you have 2 players. And comes the first player and start throwing darts on the dartboard. Okay, so it's all over the place. Okay, and finally throws the 20th shot and goes in the bullseye. After 20 shots, gets the bullseye. Okay, so that's player number 1. Okay, comes player number 2. Same thing, starts to throw, and boom, after 1 shot, hits the bullseye. So question. Do you think the 2 players are as skilled at this game? Eventually, they both hit the bullseye. But do you think they are just as skilled? Player 1 is just as skilled as player 2? No. I mean, the first one, kept on throwing darts and eventually hit the bullseye. But the second one came and just boom, the first time, it hits the bullseye. So they're not the same players. They're not as skilled, as skillful as each other. So that's kind of the thing with multiple comparison. If you keep on trying tests, eventually you're going to find a statistically significant result. Yeah?
How do you say that? Because if you ask the second person to keep throwing 20 darts, he may get the same distribution as the first person. It may have pure luck that he hit the bullseye the first time.
Okay, I missed. If say that again, can you repeat it a little bit louder?
He said the result, the fact that the second player hit the bullseye the first time
Yeah.
proves that he's a better player than the first one. But if he were to throw 20 times, just like the other guy, he may get a distribution just like the other guy.
Oh, I see what you're saying. Okay, so what you're saying is, if you really want to compare the two players, is let them both throw 20 times and then see how they differ. Because the second player, by chance, it could have been the 20th of the first player just happened to be the first of the second. Well, that's a good point. That's a good point, yeah. You're right. If you really want to be correct, let both of them shoot 20 times and let's see how they differ. So you're right, yeah. But, okay. You're spoiling my analogies. The point is that, the point is, if you do a lot of tests, and it's very easy to do that, very easy. You keep on doing things, eventually you're going to get a statistically significant result. Actually, 5% of the time, you're going to get a statistically significant result. So, when do you do multiplicity adjustment? Formally, whenever there are more than one primary endpoint or primary hypothesis. So if you have, for example, you want to, in the drug abuse field, you would say we have two important primary endpoints. We have drug use, we want to lower drug use, and we want a retention. We want to keep the patient or the participant in coming to the sessions. And that's very important. So retention is important. And you say these are equally important, and therefore I have two primary endpoints. So if you have two primary endpoints, you have two tests. Well, then you have to adjust for it. You have to penalize yourself for having too many possibilities of getting a statistically significant. You have to make it harder for you to claim a statistically significant result. That's what multiplicity adjustment does. Or, for example, you have more than two treatment conditions. So you have A, B, and placebo. And you want to compare A versus placebo, B versus placebo, A versus B. You have three tests. If it is the primary analysis, you have to formally adjust for that. How do you do it? Very complicated, many methods, many papers, many books. But you have to adjust for it. You have to make it harder to claim something to be significant because you're trying 3 times instead of just 1. That's the formally. Formally means you are using a technique that has been proven for adjusting for multiple comparisons. Informally, if you're doing secondary analysis or subgroup analysis, so you're writing a paper, you try gender differences, this differences, etc. You try different races, different age groups, and all that. And you say, well, because I've done so many, instead of using a cutoff of 0.05, I'm going to use a cutoff of 0.01 or 0.005. So this is very informal. It's not calculated. You're just saying, I'm doing too many, so I'm going to make it harder on me to claim statistical significance. But as we know, secondary analysis and subgroup analysis are not definitive answers, so that's why it's okay to do it informally. But the primary analysis, the main reason, the one reason you're doing the trial, if it has multiple statistical hypotheses or statistical tests, you have to adjust formally. So whenever you see things, and it's very easy, like I said, you can, let's try this, let's try that, let's try this, let's try that, and do it this way, and do it that way, and do it this way. Very quickly, you can get into the hundreds of tests. And then you say, oh, I found the less than 0.05. Well, 5% of the time you will, even if there is no difference. So there's the second way. Remember, there was the first way. There are two ways to get statistically significant results, guaranteed. Remember the commercial, guaranteed. You can analyze a very large sample, or you can keep on trying different statistical tests on different assessments, on different outcomes, on different subgroups. Keep on trying, and for sure, you will find something statistically significant. I guarantee it. But then what does it mean? For the first one, we talked about this. For the first one, you say, is it clinically meaningful? I find a difference of 0.01 or whatever, of 1 millimeter, but is it clinically meaningful? The person used to have 50 headaches a week, now has 49 headaches a week, and it is statistically significant. Well, is it clinically meaningful? And the second one is basically by adjusting, multiplicity adjustment, you make it harder on yourself to reject the null hypothesis, either formally or informally. Or you can just say it in the paper. You say, well, we did about 25 differences, tests, and we realized that even if I'm showing you a p-value of 0.04, we have to be careful not to interpret this too seriously. It's okay. That's informally. Okay, we're doing great in terms of time. So the bottom line key points. I have 2 slides. I'm going to list those things. This is basically a summary of everything I've covered. And if you're going to see those points, if you understand these points well, I think you have the foundation for hypothesis testing. So, statistical inference uses results from a sample, from the population of interest, to draw conclusions about the population. Okay? The null hypothesis is set up with the hope that it will be rejected. Alpha is the chance of making a type 1 error. In other words, of concluding that there is a difference when in fact there isn't. Is the chance of rejecting H0 when H0 is true. Beta is the chance of making a type 2 error. In other words, of concluding that there isn't a difference when in fact there is. Power is 1 minus beta is the chance of concluding that there is a difference when in fact there is. So it's a correct conclusion. We want the power to be high. We want alpha and beta to be low. And how do we adjust for those? It's through the sample size. The investigator controls the chance of making a type 1 error, alpha, and the chance of making a type 2 error via the sample size. If you increase the sample size, it's costing you more, but you can lower both alpha and beta. P-value is the probability of obtaining a result as extreme or more extreme than the one obtained if there were no difference, if the null hypothesis were true. That's the p-value. Remember, nothing more, nothing less. Statistical significance does not mean clinical importance. You can always find something statistically significant. All you have to do is raise the sample size. Eventually, you will get something statistically significant, but it doesn't mean it's clinically important. And that's why I didn't mention that, but or maybe I do. The confidence intervals are very helpful. Confidence intervals are very useful to better understand results. P-value is just, okay, p-value is 0.01. So the investigator says, can you tell me in English what have we found? That's where confidence interval. Then you can say, well, The difference is about 3 plus or minus 1. Oh, okay. So we have reduced the number of headaches by 3 over 1 week. Okay. Is it clinically important or not? Well, that's the expert who decides. And multiplicity adjustment is needed with more than one primary hypothesis. If you do more than one test, if it is the primary analysis, you have to adjust formally using a technique that has been, is established. How do you do that? You talk to a statistician. It's not straightforward. People know the Bonferroni. Have you heard about Bonferroni adjustment? That's the simplest method of adjusting. So it says, it's very conservative. It says if you have 2 tests, divide 0.05 by 2. So now the cutoff is 0.025, not 0.05. That's called the Bonferroni multiplicity adjustment rule. It's the simplest, but there are many more sophisticated ones that work better. But that's one way of doing it. Think about it this way. If you have 3 tests, take the 0.05, divide by 3. That's your new cutoff. You've made it harder to reject the null hypothesis. And finally, Bayesian approach is gaining popularity as being more intuitive and is worth considering. So if you're talking to a statistician, say, well, what about, can we do it through the Bayesian approach? You'll know very quickly whether the statistician is a Bayesian or not. Very quickly. And that's it. So we have 3 minutes for questions. Thank you for attending. Hope it was helpful.